will just pull out the premises themselves. Okay. If there's anything else going on in the paragraph, examples, whatever, ignore those. Okay. Yeah, good question.
Professor, I wanted to ask, how would you know, like, if you said this was the conclusion part?
Yeah.
That was the conclusion, the last part, like near the end. And then there's like a descriptive word, like so, therefore, and whatnot. But sometimes it could be like the conclusion could be like the middle or in the like beginning as well. How do we distinguish that from just like a summarizing conclusive sentence?
Great question.
Sorry, I would, to be more specific, if the conclusion was in the beginning, how would I know that that's the conclusion and not like a therefore statement towards the end? How do I distinguish?
A great question. I didn't leave enough time to go over the argument reconstruction in detail, so I'm going to upload a video, and I will answer that question among others. So how do we know when this is the conclusion and not merely, kind of summary wrapping up something like this? So I'll aim to upload that this evening, so keep an eye out. And then if you've got any questions, just come to my virtual office hours tomorrow. Yeah, everybody, let's head out to make room for the next class. Hey. What was that? Well, it's exam review today, so indeed. I have some candy. Do you want any candy? No. Okay. You passed. There you go. All right. Take care. See ya.
All right. Well, good afternoon, everyone. A couple of housekeeping things here before we continue on with our lecture. So we, of course, are in the rebuttal phase now for term quiz 3. So if you're planning to submit a rebuttal, you got to get that in by the end of the day on Friday. And then I'll try to look them over sometime on the weekend or early into the next week. I wanted to kind of remind you that our 4th term quiz comes up right after reading week. It's the Monday and Tuesday right after reading week. So I'll just mention that today and tomorrow, because I won't see you for a full week before that happens. So just so you're prepared for that. And then, I posted an announcement on Tuesday that we've extended the 2% deadline for the 2% bonus deadline for completing short communication 1. We're just going to push it right out to the November 14th date there, just to give people, I know there's lots of people who are, pushing along pretty well at this project and just need a bit more time. And, even November 14th, I'd be happy, I think the TAs would be happy too, to kind of have more early submissions than late ones. So we've extended that deadline out, so just keep trucking away. I'll just remind you that if you had a submission sitting there waiting for evaluation and it's been 3 business days, shoot me a quick email and I can have a look at it myself or try to nudge the TA either way. But we want to keep things moving in a timely fashion so that you don't get backlogged at some point. Other than that, I don't think I have anything else to say. Are there any questions about things before we continue on here? Everybody's all good? Okay. All right, so we are hopefully going to finish up this electrophysiology lecture here today, and then we'll switch gears to our next topic on synapses and neurotransmitters. We spent yesterday talking, or Tuesday, talking about action potential propagation speed. We learned that having greater axon diameter helps to speed up action potential propagation, as does myelination. And we went through and we talked about kind of the theoretical underpinnings of why that's the case. So all of that leads us today to talking next about how we classify axons within the nervous system on the basis of how fast they can conduct action potentials. And so the figure here kind of shows us some of the groups of axons, some of the classifications here. So I'll use this figure to kind of introduce you to the topic. So what they're showing us here at the very top is the classification sort of names here. And so we'll start here with what are called the A-alpha axons, or A-alpha fibers, fiber just being another name for an axon. And you can see here when you look at the A-alpha axons in comparison to all the others, the A-alpha axons are clearly the largest diameter, and they also have the thickest myelin sheath. And so because they have those two characteristics, they are not surprisingly the fastest in terms of their action potential propagation speed. And you can see here, they've pegged that speed at 80 to 120 meters per second. If we try and convert that over into something that's a little bit more familiar, like kilometers per hour, that's akin to about 360 kilometers per hour. Right? So every time I put this number up, I think about driving in to work this morning down the 401. I may be going as fast as 120 at some time. So I mean, if there were action potentials driving down the 401 with me, I mean, they'd be those guys who are flying past you at insane speeds. That would be at least the A alphas. You can yell that out next time a driver flies past you, like goddamn A alpha, right? That'll remind you that these are the fastest of all the nerve fibers. Next up here we have the A betas. So again, you can see they have a little bit lesser diameter. Their myelin sheath is not quite as thick, and you can see their speed is correspondingly slower. They don't show us the A gammas here, but they do exist. They skip over to the A deltas. Again, you see the trend here. The diameter is a little bit lesser. The myelin sheath isn't quite as thick. The speed is therefore a bit slower. There are B fibers as well, again, not shown here. They, again, just keep the trend going. They are thinner in terms of their myelination. They are smaller in terms of their diameter. They, therefore, have slower action potential propagation speeds. And then lastly, we come to the C fibers here, which I'm trying to point at. Lastly, we come to the C fibers here. You can see that they are very thin in terms of their axon diameter, and they are non-myelinated, right? They are non-myelinated nerve fibers. And so because of their very thin axon diameter, because of their lack of myelination, you can see here they have the slowest action potential conduction speed, down around 0.5 to 2 meters per second, which is akin to about 4 kilometers an hour, right? So kind of a walking speed. So there's quite a bit of a difference if you kind of put it into that perspective, right? Between A-alpha axons that are basically like cars flying down the 401 at max speed, all the way down to these C fibers that are basically people walking in terms of their speed. There's quite a bit of variation in terms of how fast these different types of axons can propagate action potentials. So the next thing I want to address here is, especially when you think about that C fiber there that's not myelinated at all, one of the things to think about is, well, again, why would we ever have non-myelinated, axons within our nervous system that are going to be transmitting action potentials as slow as they do? And of course, the answer to that question is given here now. It says, the myelin sheath requires a lot of energy to maintain. We learned in lecture on Tuesday that the myelin sheath is comprised of a living cell, right? It's like the processes of an oligodendrocyte or the entirety of a Schwann cell wrapped around the axon. These are living cells. Myelin is a living tissue, right? So sometimes people will kind of make an analogy between the myelin sheath and say the insulation on an electrical wire or something. Those really aren't perfect analogies because neither one of those things is living, I guess, the covering of the wire. But, the idea is that this insulative sheath, this myelin sheath is wrapped around the axon. It's a living tissue. It therefore requires energy to sustain it. And obviously, the thicker the sheath is, the more energy it requires to maintain it. And so it's just like we were talking about the other day about, why aren't all axons large diameter? If that's going to maximize the action potential propagation speed, it's again this idea of trade-offs, right? So there it was more of a speed-size trade-off. Here it's more of a speed-energy investment trade-off. It's not worth putting a ton of energy into maintaining a myelin sheath to have very fast action potential propagation speeds, if that propagation speed doesn't pay off in some way, right? So again, we find that we tend to invest a lot of myelin into those axons where we need the speed, and we don't have myelin for those axons where speed is not required. And that kind of leads us into this next slide here, where it's going to talk about, for both the motor and the sensory divisions of our nervous system, this slide talks a little bit about, okay, well, what kinds of fibers do we have associated with these different motor and sensory functions? And we'll be able to see from this analysis, where is the body kind of prioritizing speed and where is it not? And so if you look here on the first part of this figure, where it's talking about the motor division of the nervous system, here it talks about where do we see A-alpha axons within the motor division of our nervous system. These, of course, as we just established, being the fastest of all the axon types. Well, we see it in relationship to extrafusal muscle fibers. So we learned about these a little bit earlier in the course. These are basically just your regular old skeletal muscle fibers, right? So they're part of the somatic nervous system. So this is where we see the fastest axons within the motor side of the nervous system is they are associated with our skeletal muscle fibers. And if you look at the A gammas that come next here, it talks about them being associated with the intrafusal muscle fibers, which we know from earlier in the course are those specialized fibers that are associated with the muscle spindles, right, that allow us to detect how stretched our muscles are, right, so we don't overstretch the muscles and damage them, but also so that we know the position of our limbs, right? I can tell whether my arm is extended or flexed based on how much stretch there is of the bicep and tricep, for example. So if we put all that together on the motor side, we're basically saying, hey, where we have the fastest action potential propagation, where we have the axons that can transmit nerve impulses the quickest, it's all in relationship to our skeletal muscle fibers, our somatic division of the motor system. And so that's where we're putting our priority for speed on the motor side. And when we think about why we do that, again, I think it's because of how directly important our skeletal muscles are to our survival, right? If you think about it, if, one of you was going to throw a dart at me or something, you're like, hey, I'm going to try and, get a dart into his eye or something, so he, falls to the ground and shuts up and we just all go home. My ability to see that dart coming and get my hand up there to block it quickly is critical to my survival. That fast ability to operate skeletal muscles is so important to survival, so we can understand why we are investing so heavily into that system. By contrast, when you see the B fibers, which are, again, kind of lightly myelinated, thin fibers, so not very fast, and then C fibers, which are not myelinated at all, very thin, very slow, we basically see these slower fibers within the autonomic nervous system, right? Our parasympathetic and sympathetic nervous system, we have much less speed there, right? Because we have these B and C fiber types that are, smaller diameter, less myelinated, not as fast. So we're not putting as much priority in terms of speed into the autonomic nervous system as we are into our somatic nervous system, right? And this, again, presumably is because there's less of a need for speed in that system to promote our survival, for example, right? But also, again, we kind of, if you think about, again, maybe a threat. You think about suddenly we hear a loud banging outside the lecture hall or something, a huge sound goes off, and you think maybe you're under attack. Again, having that ability to very quickly contract your skeletal muscles to maybe protect yourself, right? That's priority number one. You being able to ramp your heart rate up really quick so you can run out of here, well, that doesn't have to happen quite as quickly. I can run out of here even if my heart is kind of sluggish because I could maybe rely on anaerobic metabolism for a period of time as I start running, and then once my heart finally gets going because I've turned on my sympathetic nervous system and activated my heart to contract faster, then maybe as I'm partly out the door, I can switch back over to aerobic metabolism. I don't need as fast a response from my sympathetic or parasympathetic nervous system in terms of promoting my survival as I need from my skeletal muscles. So we are prioritizing on the motor side speed within the somatic division, speed of our skeletal muscle contraction, way over prioritizing speed within the autonomic nervous system. On the sensory side, we see an interesting thing here too. The fastest fibers, the A alphas, they're associated with our muscle spindles, right, which we just talked about. So they are related to our skeletal muscle function. The Golgi tendon organs, which we haven't talked about, but they're basically the same as muscle spindles. They're just located in the tendons instead of being located in the muscles themselves. And then they both relate to this sensation we call proprioception, which again is just this ability to be aware of where your different limbs are in space. Like, what is your body position? Where are your limbs oriented relative to your body? So even on the sensory side, we see again that we're prioritizing, this sensation that is directly related to our ability to contract skeletal muscles in an efficient way, right? So the totality of what we're seeing here is that, as humans, we are investing a tremendous amount of speed. We're investing in speed, especially when it comes to our ability to contract and operate our skeletal muscles in a very fast and efficient way. The A beta fibers, which are a little bit slower here, you can see that they are associated with touch, pressure, and vibration. So we have pretty fast fibers associated with touch, pressure, and vibration. And then you go down to the A deltas and down to the C's, which are both a little bit slower, especially the C's, of course. And there's where we start to see pain and temperature kind of come in. And so what's very clear is that we're prioritizing speed in terms of touch and pressure and vibration well over temperature and pain. And again, the reason for that is because of the survival implications, right? I mean, if you imagine that someone is touching you, you're probably at greater risk of some survival, quandary compared to if you're just cold, right? I mean, if you're cold, okay, you're probably not going to immediately die, right? So you have time to kind of move and find some place to get warm. But if there's someone grabbing you, then ultimately you're probably at a much greater threat risk. So we prioritize, again, in terms of speed, touch, pressure, and vibration because all these things are more directly a threat to us. Think of some big elephant came barging into the building here. You'd feel the vibrations of it stomping around, and you could feel that and respond to it quickly and know maybe you're in a threat. Touch, pressure, vibration, all more threatening stimuli that we need to respond quickly to compared to temperature or pain. Even pain, we don't need to be aware of pain rapidly, necessarily, right? I mean, we need to eventually be aware of pain so that we know, hey, we're injured and we have to try and deal with that injury. But I don't need to be immediately made aware of pain, especially, again, in comparison to what is really a more threatening stimulus, touch or pressure or vibration. But I also want to point out here, this is really interesting too, again, you have these pain fibers that are these A deltas, so kind of fast. And then you also have these other pain fibers that are C fibers, so very slow. This difference in these two pain fiber types has been typically used to explain the two different kinds of pain that people experience, like that sharp acute pain and then like dull chronic pain. So if you imagine, for example, that you were to cut yourself, right? You accidentally, nick yourself while you're cutting onions or something. You immediately feel that like sharp pain right away because you've cut yourself. But then for a long period afterwards, there's like that dull aching pain that you still feel, right, long afterwards. It's generally accepted that the A delta fibers, right, the A delta pain fibers, they're probably responsible for that like sharp acute pain that you feel right away because they propagate pain sensing information very rapidly. And the C fibers, which are much slower, are probably responsible for that more kind of chronic, longer lasting pain that you experience. They send signals back to the brain more slowly, and so that kind of second pain that you feel, that more kind of chronic pain after the fact, is likely due to the C fibers finally having sent their pain information back to the brain. So that's what we currently believe anyways. What I also want to showcase here is not only do these different types of axons propagate action potentials at different speeds, they're also differentially sensitive to stimuli as well. And so that is what I'm going to show you here in this figure. So on the right-hand side here is just the setup for how you measure the data I'm going to show you. Anybody that's done B32 here, the setup will seem familiar because you've probably done stuff with the crab nerve, for example, in that course if you've taken it. If not, then let me just walk you through what we have here. We have a piece of a nerve here in yellow, and on one end it's connected to a stimulator, so we can basically stimulate it electrically. And on the other end, you have a recording electrode that's basically monitoring the membrane potential, so we can see action potentials occur. And then that recorder is connected to an amplifier just to make the signal easier to see. And then you can ultimately see how that's connected to a computer, and especially a computer monitor, so we can just visualize the results. And so then here are the results on the left-hand side. So you have these four different plots they have here, one on top of the other. You can see that in every plot, we're going to apply a stimulus at time 0, and then we're going to see what happens within this fiber. How does it respond? And you'll notice that they tell us that at the top, we have the weakest stimulus, and then as we go down the plots here, we are increasing the stimulus strength. So if we look at the very top plot first, what you'll notice here is that when we apply a very weak stimulus to this nerve, we see one kind of response here. That's it, right? And it says that those are the A alpha fibers. So what that tells us is the A alpha fibers, we already know they're the fastest of the fibers, but they're also the most sensitive to stimuli, right? Because they're the only fibers that have actually responded to this weak stimulus, so they must be the most sensitive to that stimulus, right? And you can see here, too, how quickly we apply the stimulus again at 0, right? And then that stimulus leads to a response that we're detecting way down there at the other end of the nerve, within a millisecond, right? So we're transmitting, those A alpha fibers are transmitting their action potential very rapidly across the length of this nerve. If we go to the second plot here, so we're applying a little bit stronger stimulus now, you can see that 2 peaks show up, right? So the A alphas again appear, but now another peak comes up, which is the A betas. So the A betas are the second most sensitive. So we're starting to see this kind of relationship between speed and sensitivity. They seem to be directly correlated. You'll also notice here, of course, the A betas being a little bit slower. That's why they show up to the recording electrode just a little bit afterwards, after the A alphas did, right? The signal doesn't travel as fast through the A beta fibers, so it doesn't get to the recorder as quickly, and that's why that peak shows up a little bit later. You go on to the third panel, and now you have 3 peaks showing up when you apply a slightly stronger stimulus. Your A alpha, your A beta, but now your A gammas show up, right? So they're the next most sensitive. And again, you can see how the A gammas, being a bit slower than either of the other two, again, their peak doesn't show up until later. It takes longer for the signal to travel down those A gammas to get to the recording electrode, so it shows up a little later. And then in the final plot here, they just kind of give up and just give it the max stimulation here. They don't want to go through piece by piece. But you can see now, the A deltas come on board now, the B's come on board, the C's come on board. So as you keep adding more and more strength to the stimulus, you keep recruiting these, slower and slower nerve fibers. So again, that direct relationship between propagation speed and stimulus responsiveness is clear. But again, we can see here how the A deltas and betas, again, both being slower than the A alpha, beta, gammas, their peak shows up a little bit later. And then the C's, being not myelinated, very thin, very slow, their peak shows up way later. And again, there's almost a 50 millisecond difference there in terms of how fast the A alphas are transmitting action potentials relative to how fast the C fibers are. Right? So again, we can appreciate that there are different types of axons. They propagate action potentials at different speeds. They have this directly correlated difference in their sensitivity to stimuli. And we can then again see from the figure above, the table above, that we are putting these different types of nerve fibers in different locations within the nervous system, prioritizing speed, especially in our skeletal muscle and movement, and things like touch pressure and vibration, and not investing as much speed into the autonomic nervous system or to pain or to temperature, right? Because we can't maximize everything. Otherwise, we'd have too many large fibers, too much expense in myelination. We have to make some decisions.
For like C and the delta and B, the stimulus is like spread out on a large area. Is that like positive for certain applications of the body? Like,
Is it?
Imagine like you give like one stimulus that gives us like a response that's
like a prolonged response. Yeah. Which I think, again, is kind of like going back to the idea of the chronic pain. Yeah, you have this much longer responsiveness to that pain. I mean, the reason you get that spread there, I think, is because you've got some variation. Like the C fibers, for example, probably aren't all one uniform diameter. So if they're all a bit of variable diameters, then they're all going to transmit signals at variable speeds. But you're right, that could lead then to that one pain signal basically coming back to the brain over a prolonged length of time because of that variability, which then increases the chances that you're also going to respond to it. So yeah, there could be value in the spread for sure, could have value here in terms of it being like a repeated stimulus, one stimulus leading to repeated signals coming in at different times to the brain. Almost like a temporal summation kind of a phenomenon. Yeah, that makes sense. Yeah. Okay, the next thing I want to talk a little bit about here is myelination. And we already know that myelination speeds up action potential propagation. I want to talk a little bit about how, some factors that can affect the degree to which nerve fibers are myelinated. And so one of the factors I'll talk about first here is age. And so I have this plot here where they're showing us age on the x-axis, and then they're showing us on the y-axis something called fractional anisotropy, which is basically a measure of how much myelination is present. Now, let me just kind of walk you through very briefly what fractional anisotropy is. How do we actually use it to measure myelination? Like, how are these things related? So the way we measure fractional anisotropy involves an MRI-based technique. So most of us are familiar with the idea that MRI can be used for, say, brain imaging, right? And so on the picture on the right here, it says they're showing us a standard MRI image, and we would do an MRI of someone's brain to maybe look at the structure of the brain. And you could use MRI on other parts of the body, too, to look at the structure of other parts of the body. but that's how we're most familiar with MRI, as kind of this structural visualization technique. We can see the structure of something. With fractional anisotropy and trying to measure the degree of myelination, you're using a slightly different kind of MRI technique called diffusion-weighted MRI. And so unlike the standard MRI, where it's like we take one image, basically, and see what the structure looks like, with the diffusion-weighted MRI, you're taking a series of images, and you're kind of comparing how those images change over just very brief periods of time. And what that allows you then to actually be able to see is how are water molecules within the brain, which is what MRI basically sees, how are these water molecules within the brain, how are they moving within the brain? So we're just letting them kind of move around as they will and taking multiple sequential MRIs to see how are these water molecules moving around within the brain. And then the question is, well, how does that tell us anything about myelination? Well, it's because if you imagine that there are water molecules, and there are, you don't have to imagine it, there are water molecules present within the cytoplasm of an axon. If that axon is heavily myelinated, those water molecules are going to be forced to travel down the length of the axon, and they'll be unlikely to travel across the axonal membrane. So what we should see then in the MRI, if we follow water over time, is an area that's heavily myelinated. The water molecules will all be moving in kind of one direction. Right? They'll all be moving in one direction, kind of down the lengths of axons. And so we'll be able to see that on the MRI image. We'll be able to see that the water molecules have moved in kind of one constant direction because they're caught within these myelinated axons and can't easily cross the axonal membrane. But if you have a region of the brain that's either not myelinated or a region that has damage to the myelin sheath or just, for whatever reason is not quite as myelinated, those water molecules might go down the cytoplasmic the axonal cytoplasm, or they might now be able to cross the membrane to leave the neuron, so they could therefore be moving in all kinds of directions. And so if we have our MRI and it shows the water molecules are moving in all kinds of different directions and not kind of in one uniform direction, that would be indicative of a lack of myelination in that region. So that's how we can measure fractional anisotropy, and basically the higher the fractional anisotropy value is, that corresponds to more water molecules moving in the same direction, and that means you've got higher degrees of myelination. Right? So high fractional anisotropy means high degrees of myelination. So with that knowledge, we can go back to our figure where we're looking at age and fractional anisotropy, so our measure of myelination, and we see here that they looked in this study at a whole bunch of different brain regions. So they're all color-coded here, the different brain regions that they examined in the study. But what you notice is that basically for all these brain regions, the peak myelination happens right around 20 to 30 years old. That's when you have peak myelination in your brain. So the majority of you are kind of either in that range or right there on the edge of that range. You guys are pretty much at peak myelination right now. Right? Your brains are going to be as fast as they're ever going to be in terms of processing information, responding to stimuli, and you've probably got 10 more good years with that. And then you notice that once you get beyond 30, it's all downhill from there, right? So, unfortunately me, sitting here at 43 on this part of the curve, I'm well past peak myelination, right? And I know it, and you probably know it on days when I'm standing here thinking of a word for 10 minutes, right, that the processing speed just isn't what it used to be, right? And that's a natural phenomenon, right? We get, it increases with age, as you can see, right? Levels off of that peak at 30 and 40 and then gets worse over time. So, you are in peak myelination, take advantage of that, right? This is your time to use your brains to the best of their abilities, because, once you get past 30, the decline begins to set in. So we know that age is one factor determining the degree of myelination and therefore kind of how fast action potentials can propagate. Another factor, not surprisingly, is really how much you use your brain, right? This is I grabbed this from I think a poster somewhere anyways that someone was presenting, and basically they showed that more practice led to more myelination. So I'll walk you through the study a little bit more detail here. So they were looking specifically at people playing piano. All right, so it talks about adolescents and adults. So they basically found a bunch of adolescents and adults who play piano, and they measured the degree of myelination in 2 regions of their brain. So the first region was the corpus callosum. This is the connection between the 2 cerebral hemispheres, right? So we have to send information back and forth between our 2 cerebral hemispheres, and that's really important, especially for functions that require the use of both sides of the body in a coordinated fashion, like playing the piano. I don't play the piano, but even I know that I need, ideally, if I'm a good piano player and not someone who's just trying to play chopsticks with one hand, I need to be able to work both hands in a highly coordinated fashion in order to play the piano. The corpus callosum then allows for, both hemispheres of the brain to communicate so that they can control the movements of both sides. And the faster that communication is happening, obviously the more coordinated your movements are going to be, and the better a piano player you're going to be. And in fact, you can see here in this study, they showed that how myelinated the corpus callosum was in these piano players was positively correlated with how many hours of their life they had spent practicing, right? So, again, and look at the number here. It's 10 to the power of 3 hours, right? So, some of these people had practiced 6,000 hours, right? I don't know how much that translates into in terms of days. I'm on the top of my head here. My processing speed is too slow, but that's a lot of fucking time spent playing a piano practicing, right? So, long story short here, if you want to get good at something, you have to work at it, right? And your brain will actually respond, right? That those parts of your brain that you're using with that repeated practice, your brain starts to say, hey, it's worth investing in myelinating these brain regions because that speed is actually going to benefit you. That energy cost you're putting in is actually going to yield benefits in terms of you being a better piano player. The other brain region they looked at here is something called the arcuate fasciculus. This is a part of the brain that sort of translates written things into motor output. And so in the case of playing piano, this brain region would be involved in you seeing the musical notes on a piece of paper, or piece of paper, but you seeing the musical notes on the sheet music, and then being able to, coordinate that with the movement of your fingers to play those same notes on the piano. So another brain region that's very important for piano playing, and again, we can see that its myelination increases as a positive correlation with practice. So again, we see that the more effort we put into doing something, right, the more we're using a brain region, the more the brain says, hey, let's put myelination into that region. It's going to cost us energy, but it's worth it. But of course, the opposite is also true, that if you don't use a certain part of your brain, likely it will undergo demyelination, right? And you'll just get worse and worse and worse at that particular function. So as is so much the case with the body, it's use it or lose it, right? You use it, and the brain responds by saying, I'll myelinate this more. If there are parts of the brain you're not using as much, the brain says, hey, I'll take away the myelination here, because it's just not worth the energetic investment to put that myelination there. Last thing I want to talk about in this lecture topic is to talk about demyelinating disorders. So what happens when your myelin sheath gets lost? And so as it says here in the top right corner, there are 2 major demyelinating disorders. There's multiple sclerosis, which most people are fairly familiar with. This is a demyelinating disorder of the central nervous system. But then there's also Guillain-Barré syndrome, which is a similar disorder, a demyelination disorder within the peripheral nervous system. Right? And so they share a lot of similarities. What I want to do is I want to walk through kind of not so much what causes the demyelination. There's always a lot of question marks around that still. Mostly, especially with multiple sclerosis, most of the evidence points towards it being autoimmune, right? So there's probably some myelin protein or some myelin antigen that the immune system mistakenly attacks and destroys the myelin. But there's still enough of a question mark there that I don't really want to deal with it. What I want to focus on is just what happens after the myelin sheath has been destroyed, right? And so obviously the first thing you'd say to yourself, okay, well, if I experience demyelination, right, of a myelinated axon, so sections of the myelin sheath are being torn out or destroyed, the first thing you'd expect, and this is what happens, is action potential propagation will probably end up stopping for this neuron now. Because you have lost the myelin sheath in a given region, and so as the sodium ions are now flowing through this demyelinated region, they're very prone to just leaking out now because there is no myelin sheath to resist that, and they may not make it down to the next node of RANVA before the potential goes back below threshold, so then you can't trigger action potentials down further within that neuron, and ultimately now this action potential just doesn't get propagated down the length. You may have a difficult time doing things like stimulating, muscles to contract, for example. And yeah, a lot of people who have multiple sclerosis, they go through these periods of time where, they're trying to do something. They're trying to contract some muscles, and they just can't actually execute because the action potential is dying now as it passes through these demyelinated sections. However, the neurons are clever. They do try to solve this problem over time. So what they will start to do is they will start to increase the expression of voltage-gated sodium channels within the demyelinated region. And so as they start putting these voltage-gated sodium channels into the demyelinated region, this now allows for action potentials to be generated within that demyelinated region in that kind of continuous conduction fashion that we talked about yesterday. And although this is going to be slower than if you had the myelin sheath, it's better than having nothing, because now you can generate action potentials in this demyelinated section. You can hopefully get your action potentials to propagate through that section now and either get back to another myelinated section, or maybe you just have to do continuous conduction the whole damn length now to get it down to the end, to the terminal. Either way, at least now you've restored action potential propagation. And so this leads to the remission phase of multiple sclerosis, where people initially had symptoms, and then suddenly the symptoms disappear because the neuron has responded in this way, right? And even though, again, the action potential propagation is slower, at least it's been restored. However, this is kind of a transient fix, okay? Because what happens, and we haven't yet talked about this player here, there's a protein, which, again, we haven't yet talked about, called the sodium-calcium exchanger. And as its name would suggest, its job is to exchange sodium and calcium across the membrane. The way that it typically functions, okay, in a healthy human being, healthy cell, the way it normally functions is we've got our sodium-potassium ATPase pumping sodiums out of the cell. The exchanger allows those sodiums to come back into the cell in exchange for calcium leaving the cell. And so the calcium-sodium exchanger here kind of works hand in hand with the calcium ATPase to get calcium out of our cells. Right, we talked about the calcium ATPase earlier in the course. So these two things work hand in hand to ultimately get calcium out of our cells and keep the intracellular calcium level pretty low. But what happens here in this remission phase of multiple sclerosis or Guillain-Barré syndrome is that you've still got your calcium-sodium exchangers there, but they start working in reverse. The reason is because now you've got so many voltage-gated sodium channels in this demyelinated section, letting so much more sodium into the cytoplasm of this neuron than when it was myelinated, that you have to turn these calcium-sodium exchangers around to start getting all that sodium back out that's coming in. You don't have the sodium-potassium ATPase capacity to deal with this here, so you're using the exchanger to say, okay, I got all this sodium coming in because I've got all these new voltage-gated sodium channels I put in here to restore action potential propagation. All this sodium is coming in and I have to get rid of it, and so the exchanger works in reverse. It starts getting the sodium out but brings calcium in its place. And the problem with that calcium influx is when calcium levels in the cell get high, and especially for any chronic period of time, that actually leads to apoptosis. It leads to proteases being turned on. It leads to phospholipases being turned on. It leads to the cell eating itself from the inside. So we're going to talk more about this in the synapse and neurotransmitters lecture too. But whenever calcium levels get too high within a cell, within the cytoplasm of the cell, right, that basically is a cell death signal. And so, yes, you had this remission phase where things were good, but then what happens in the long term is as all this calcium now starts to come into the axon, ultimately that leads to the death of that axon. And so you go from, I've got symptoms to remission to now symptoms again, right? The initial symptoms were due to the demyelination and the inability to propagate action potentials. The remission was brought about by increasing the expression of these voltage-gated sodium channels to try and still allow for action potential propagation to occur. But then the final set of symptoms is reflecting how that remission phase and the expression of those voltage-gated sodium channels ultimately leads to this intracellular influx of calcium that causes apoptosis and damage to that neuronal axon. And then once the axon gets damaged, ultimately you can't propagate action potentials anymore. And so now the symptoms are going to be kind of permanent at that point. Okay, so that ends that. Still got 10 minutes, so I'm going to at least start the synapses to get through just the little, kind of dull first parts. If I can figure out where my cursor went to. There it is. And then that way tomorrow we're past the little dull introduction parts. And we can hopefully talk about cooler things. So yeah, just a few minutes here just to kind of get us started on this topic, kind of a foreshadow for tomorrow. So yeah, our next big topic, and it's our final nervous physiology topic. And I'm hopeful, it's only week 8, so I'm hopeful that we'll have a good couple of weeks at the end where we can talk about the cool stuff related to reproduction. Our last nervous physiology topic is synapses and neurotransmitters. This, again, I think I mentioned before, is a topic I had pulled out of the course for a couple of years, and then apparently it was causing all kinds of chaos at the D-level courses where you need to know this information, so it's come back. Okay, so we start with synapses, and we already know a lot about synapses, so again, this is just nice, light review. The synapses, of course, the word synapse, as it says here, comes from Greek, and it literally means to join together. And so what do synapses do? They literally join neurons together. And so you can see that pictured here. We've got 2 neurons, and the place where they are connected together is called the synapse. Now, when we are looking at the 2 neurons that are joined together by a synapse, one of those neurons will be called the presynaptic neuron. That will be the neuron that is sending nervous communication out. And then the neuron that is receiving that nervous communication will be called the postsynaptic cell. So you'll hear these terms a lot. Pre- and postsynaptic neurons, pre- and postsynaptic cells. The presynaptic neuron is sending out the nervous signal, and the postsynaptic cell is the receiver. It's being stimulated by that signal. Now, of course, what we have to appreciate is that, a given neuron can be either presynaptic or postsynaptic, depending on the particular synapse you're looking at. So again, when we're looking at this synapse here, right, this is clearly the presynaptic cell, and this is the postsynaptic neuron here. But if we imagine that there was a neuron up here, kind of synapsing over here with this neuron, well, now this neuron would be a postsynaptic neuron if we're thinking about that particular synapse. So the terms pre- and postsynaptic neuron or pre- and postsynaptic cell, they are relative to the particular synapse that you are looking at. And so all neurons are pretty much going to be, most neurons will be pre- and postsynaptic, just depending on which particular synapse you're actually looking at. We also have to appreciate that in the peripheral nervous system, the postsynaptic cell could be a neuron, but it could also be a muscle cell or a glandular cell, because we know that that's what the peripheral nervous system does, right? It communicates with those effector cells, the muscles and glands of the body, to contract them or inhibit them or make them secrete or whatever. So we could have a synapse between a neuron and a muscle cell, and the neuron would be then the presynaptic cell, and that muscle cell would be the postsynaptic cell. And then the other thing we have to appreciate here is that the picture that I originally showed here is a little bit misleading because it makes it look as if there's like a physical connection between the pre- and postsynaptic neuron. But as we already talked about earlier in the course, and I just wanted to remind you about it here, that's not the case. As this little figure shows us here, here's the axon terminal of our presynaptic neuron, and here is, say, the dendrite or cell body of our postsynaptic neuron. I guess here, I don't know why I'm pointing at the words, here, and we clearly have this space that is between them. And we call that space the synaptic cleft, right? So there is no physical connection between the pre- and postsynaptic neuron. You always have this fluid-filled space, and I should emphasize it's a fluid-filled space, right? It's going to be filled with interstitial fluid. Always these pictures make it look like it's air-filled or something, but it's a fluid-filled space filled with extracellular fluid, filled with interstitial fluid. And what we have to appreciate, therefore, is that the nervous communication that is happening between two neurons, for example, that communication has to happen across that cleft, right? Whether we're talking about the diffusion of neurotransmitters, whether we're talking about the flow of sodium ions from one neuron to another, whatever the mechanism of transmission, you have to get across that synaptic cleft. I want to introduce you at this point to, again, just some terminology for the different kinds of synapses that you find within the nervous system. So here we have a couple of presynaptic neurons, and then down here, actually we have 3 presynaptic neurons here, and this one postsynaptic neuron here. They highlight for us a few different types of synapses. They give us a few different names to think about here. So the first is the axodendritic synapse. So this is where the presynaptic neuronal axon synapses with the dendrite of the postsynaptic neuron. So we have an axon synapsing with a dendrite, so hence axodendritic. Here they're highlighting for us something called an axosomatic synapse. So here the axon of the presynaptic neuron is synapsing with the soma, the cell body of the postsynaptic neuron, so hence axosomatic. Those are the synapses that people most often think about. But here's another one which we're going to go into more detail about later, the so-called axoaxonic synapse. In this case, the axon of your presynaptic neuron is synapsing with the axon of the postsynaptic neuron, hence axoaxonic. And we're going to go into more detail about this a little bit later in the course, but this kind of synapse is largely to allow that presynaptic neuron to directly control how much neurotransmitter gets released from the axon of that postsynaptic neuron, right? It'll actually, again, we'll go into the details of this later, but this kind of synapse is really about, this presynaptic neuron being able to either increase or decrease how much neurotransmitter gets released from the axon terminal of that postsynaptic neuron. Are you okay over there? Yep. Okay. Just want to make sure. I don't want your death to be recorded. I should wander over here so the camera gets over here more. Okay, so those are the 3 kind of main ones here. I'll introduce you to the last one, which is not on this figure, and then we'll pick this up tomorrow. I want to introduce you to an interesting kind of synapse called the AUTAPS, which stands for, or basically is derived from, the auto synapse, which basically means it's a synapse between a neuron and itself. And so here you can see we've got these 2 neurons, one in green, one in blue. They're telling us the green is the presynaptic and the blue is the postsynaptic neuron, and you can see a synapse here. So this blue neuron can be excited or inhibited through this, this usual axodendritic synapse that we see here highlighted as a synapse. But the really cool thing here is that this blue axon this blue neuron has an axon that then has this part that kind of curves back and it forms a synapse with itself, with its own dendrite. So that part of the axon that turns around and goes back towards the dendrite of the same neuron, that's called a recurrent collateral axon, right? And so it's an axon that, again, just kind of recurrent means to go back. You're basically turning around, going backwards, and synapsing with yourself. And so this blue neuron can be excited or inhibited both by the synapse, so from that green neuron, but it could also be exciting or inhibiting itself through this AUTAPS, this autosynapse. And so I'll leave it there. We got through the terminology, but tomorrow we'll come back and talk about these AUTAPSes and like, what are they used for? Under what circumstances would we see AUTAPSes? Because, we used to think of them as just like wiring errors in the brain, but we now have ample evidence that they're actually really important structures. So we'll talk about them tomorrow, and enjoy the rest of your day. I'll see you tomorrow.